All right, I think we'll go ahead and get started. We have quite a few participants joining us from outside of in person, and it's really nice to see a large group here joining us today too. So I have the pleasure today of welcoming you all here and thanking you for attending. And I'll start with a little bit of housekeeping. So first, thank you for joining us for our distinguished lecture with Dr. Stuart Mostofsky. Our distinguished lecture series happens once a month from October through June. We have a fantastic series this year with a lot of amazing guests from around the world, providing you with the latest scientific updates for autism spectrum disorder. Now, a couple more housekeeping items. This lecture, we're very proud to say it's supported by the UCLA Center for Autism Research and Treatment, the UCLA Tarjan Center, the UCLA Intellectual and Developmental Disabilities Research Center, the UCLA Brain Research Institute, and the UCLA Can Reach Training Program. So thank you to all those programs for the support. For our attendees on Zoom, I'll ask that you can ask questions about the lecture utilizing the Q&A window, and we'll also take questions from the audience at the end of the talk. For your information, we'll also be recording this talk and we'll post it to our website as well. So now I have the pleasure of introducing Dr. Stuart Mostofsky, and I'm particularly excited to welcome him today as a fellow child neurologist visiting us here and also a fellow studier in the motor domain, which we'll be hearing a lot about today. So Dr. Mostofsky is a research scientist and director of the Center for Neurodevelopmental and Imaging Research at the Kennedy Krieger Institute. He's also professor of neurology at Johns Hopkins University School of Medicine. Dr. Mostofsky received his medical degree in 1990 through the Rensselaer Polytechnic Institute. I've always had a hard time saying RPI, I have to be honest, even living on the East Coast, and the Albany Medical College 6-year program. So to just note, he finished both his undergraduate and medical degree in 6 years. Not surprising that he also won the Jack Splitney Prize for Exceptional Achievement in Pediatrics. Dr. Mostofsky then went over to Minnesota, where he did his internship and residencies in pediatric neurology at the University of Minnesota, and then came to the Kennedy Krieger Institute in 1995 for a fellowship in the Department of Developmental Cognitive Neurology and stayed on as a pediatric neurologist with subspecialty training and experience in behavioral neurology as it applies to the study of childhood developmental disorders. Dr. Mostofsky, as we noted now, is the director of the Center for Neurodevelopmental and Imaging Research, and he really focuses on understanding the neurological basis of many neurodevelopmental conditions, but specifically 3. One in which he'll speak about a lot today, which is autism, but also attention deficit hyperactivity disorder and Tourette syndrome. Dr. Mostofsky's clinical practice focuses on serving children in autism, and he's also served as the medical director of the Center for Autism and Related Disorders at the Kennedy Krieger Institute. He's a member of several scientific societies, including the American Academy of Neurology, the International Neuropsychological Society, and the Society for Behavioral and Cognitive Neurology, just to name a few. I'll go ahead and focus on what, as I noted, he really has expertise around many areas, but research focusing around autism, ADHD, and Tourette syndrome. I'll focus on describing some of his autism-related work here today, which has been funded by the NIH, NINDS, and NIMH, the Autism Speaks Foundation, and the Simons Foundation for Autism Research Initiative. Dr. Mostofsky's research with children with autism is focused on careful examination of motor function, as increased insight into brain mechanisms underlying this disorder might be gained from careful consideration of the motor signs associated with autism, as many of us know have been described from the first cases of autism. Dr. Mostofsky has made many important contributions to identifying the common factors underlying motor impairments in autism and how anomalous patterns of motor learning in autism may contribute to the impaired social and communicative deficits that define the disorder. He's now leveraging these findings to develop novel game-based intervention strategies for children with autism. And as a researcher in this area, I can really say Dr. Mostofsky's work led the area of understanding some of the neural correlates or imaging correlates of motor dysfunction in autism. As noted, he also works in pinpointing neural systems affected in ADHD, understanding these impairments, and also understanding now how the efficacy of novel movement-based intervention strategies, including mindfulness practice, can target some of these areas. And his work has also looked at Tourette syndrome. Specifically, his findings have discovered with Tourette syndrome that reduced GABA-based inhibition in the brain's supplementary motor area is associated with both increased severity of Tourette syndrome symptoms and a reduced ability to actively suppress the tics. And again, in this area, impressively, his findings will provide a basis for helping children with Tourette syndrome reduce tics and sensory urges by targeting GABA and related brain signaling. So that's really mentioning many of his scientific accolades, some of the things we'll hear about today. I'll just have to mention quickly what you won't find out from his bio, but I had the pleasure of finding out very recently is Dr. Mostofsky is also quite a music aficionado, both curating music and collaborating different musicians and coming together and creating really new musical endeavors and brainstorming new musical endeavors. And I can say it started as a young age, as I found out, probably even before this, but with his bar mitzvah, which was Beatles themed. So I think a nice aspect of really bringing together science and the arts as well. So Dr. Mostofsky, it's my pleasure to have you here with us today, and thank you very much.
Thank you. Thank you for the very kind introduction and the invitation. I'm really excited to be here. I've long admired the work that many people in the center do, and it's a real pleasure to be here and really appreciate the invitation. I guess if you didn't think I was maybe had some aspects of the spectrum, maybe now that you found out I had a Beatles bar mitzvah, that just sort of seals the deal. All right, so I'm going to get right into it. I have to do some technical things here real quick. Okay, so as mentioned, I'm going to talk on action in autism. I've titled this talk "How Moving from Lamppost to Computer Vision Can Help Address Heterogeneity and Improve Clinical Care." That seems maybe like a bit of an obscure title, but I think you'll understand what I mean towards the end. Those who were at dinner last night understand entirely what I mean by all this. Oh, I forgot I got to do this. All right. So I'm going to start with a lot of acknowledgements and this somewhat now outdated pre-COVID picture of people in our lab. I don't know how to stop that, and I apologize for that. That's going to come in every now and then. I should close something, but I'm not going to be able to. I'm sorry that there'll be an occasional ding every now and then from my email. So this is really a highly collaborative work. I've really enjoyed collaboration. My advice sort of top down, so to speak, as a full professor now to all young scientists and young individuals throughout the world in ways is collaboration is key. This really, the work I'm going to present reflects collaboration with all of these people in centers and others beyond it. As mentioned, the funding has come from various foundations and NIH, but also more recently NSF as well. So I'm sorry that this keeps happening. So I like to start these talks with my favorite animal in a way. I mean, my favorite spirit animal is a horse. I'm a quadruple Sagittarius and I was born in the year of the horse. How could I not like the spirit animal, the horse? But I like to talk about the amoeba, and the reason I like to talk about the amoeba as a creature is it really sort of very simplistically explains what is life and what is the difference between living things and non-living things, and how everything from there is built up from there. As we all learned in, I remember, I think it was 7th grade biology for me, that the amoeba, oops, I did wrong there, that the amoeba does one of two things. It sends pseudopods, well, 3 things, as pointed out correctly last night. One of 3 things. It sends pseudopods out to get some kind of substance that it wants to envelop, a food substance, so to speak. It moves its pseudopods away from something that it needs to move away from, or just does nothing at all, stays still, which in some ways maybe is the hardest thing of all. And we are no different in a lot of ways, and all of biology is basically built up from that. We reach towards food substances. Right. That does work. We move away from danger. We also use these actions, these movements, to engage in higher order actions. Social interaction, which I'll obviously talk quite a bit about. I always like to point out chess from a cognitive standpoint, because if you stop 100 people on the street and say, is chess say a sport, you'll get some people say it's a sport. But if you say, is it a physical activity, or is it a cognitive mental game? Everybody will say it's a cognitive mental game. But if you think about it, as you look at Garry Kasparov, older picture of Garry Kasparov here playing chess, then you think about it, it really is what you're doing in your mind is you're mapping a series of movements. You're mapping a series of actions. And this is really the importance of what the amoeba points out, sorry about that. The amoeba points out, I'm going to keep apologizing for that. I apologize for apologizing. What the amoeba points out is that basically this is the difference between something living in like a table that, you know, it's not living. We can dynamically interact with our environment, and it's through a series of actions, regardless of what those interactions are. So the Daniel Wolpert, a great computational motor scientist, gave a TED talk where he really clearly explains this. And the title of the TED talk is The Real Reason for Brains. And so I'm just going to play a quick clip from this. Sorry, I'm having trouble with my mouse right now.
Why do we and other animals have brains? Not all species on our planet have brains. So if we want to know what the brain is for, let's think about why we evolved one. Now, you may reason that we had one to perceive the world, or to think, and that's completely wrong. If you think about this question for any length of time, it's blindingly obvious why we have a brain. We have a brain for one reason and one reason only, and that's to produce adaptable and complex movements. There is no other reason to have a brain. Think about it. Movement is the only way you have of affecting the world around you. That's not quite true. There's one other way, and that's through sweating. But apart from that, everything else goes through contractions of muscles. So think about communication, speech, gestures, writing, sign language. They're all mediated through contractions of your muscles.
So he makes this point really clearly. He goes on to give this great example that others have given too of the sea squirt, this animal that wanders around the ocean, its juvenile life and moves around. And then it's an adult life where it reproduces, it fastens itself to a rock. And the first thing it does is eat its entire nervous system. So it no longer has a nervous system. And so it digests its nervous system from the inside. And then like Connie has a face of fear, but then it eats its entire nervous system because it no longer needs to move. But it's still alive and it reproduces. And so this is the point about the basis of which, you know, we sort of build up these notions that, okay, if we look at action in developmental cognitive behavioral disorders, it might give us a window into what maybe underlies the entirety of the processes that define the disorder. So we know that autism is defined by impairments in social interaction, communication, stereotype repetitive patterns of behavior, but we also know, and I knew this, you know, my child neurology residency became imminently clear that parents would bring children, their children with autism, and I'd evaluate them. And there was constant discussion of difficulties that they had in motor domains. Now, a lot of these some of these can become obviously in sports related activities. One might think that, okay, there's a social aspect to it, but there really clearly is, and it's now been well established that there is a very high prevalence of motor skill deficits in autism. I'll talk about the term dyspraxia in more detail in a little bit. And that the idea, as sort of Daniel Wolpert laid a foundation for, that there is this sort of interaction between what maybe underlies the motor skill deficits and the core deficits of autism. And in part, that has to relate to, we're talking about a developmental process here. So learning and acquisition of these skills through motor skill procedural learning, that then some anomaly in that then could lead to impairments in motor skill deficits, as well as then quite possibly, and I think I'll try to make a good case for this, the impairments in social interaction, communication, and also repetitive behaviors. So then the question could be asked, well, okay, that's all very interesting, but why not really focus on and study, why, Stuart, did you go and turn towards motor skills? Why do you think that really is an opportunity? And in part, it's because, as I mentioned, that just in the last slide, that this motor procedural learning may be crucial to the development of a wide range of skill behaviors, including social skills and communicative skills, and therefore might serve as markers for deficits in parallel brain system important for social development, cognitive development. But also because the motor examination offers an ease and precision that's not really achievable with more complex behaviors. The motor measures can be more precisely defined and reliably produced quite often than these more complex behaviors. And also the anatomic and physiologic basis of motor control is very well delineated beyond that of the more complex behavior. So in the rest of this talk, I'm sort of going to talk about behavior quite a bit, brain associations as well, and then how this maybe provides a platform for intervention. So we go back to this slide, which I'll return to a number of times. And I'm going to start with focusing on the end here, the sort of motor skill deficits, and then I'm going to move into learning processes after that. And throughout, talk about the associations with the higher order behaviors. So dyspraxia. What is dyspraxia? What is praxis? So Hugo Liepmann in 1900 first described praxis sort of in sort of a similar case to Broca describing aphasia, where he observed abnormalities after brain lesions and described this impairment, this acquired impairment in that case, and ability to perform skilled or learn actions, despite the fact that these people could still engage in simple sensory motor behavior. The core of it really is ideomotor praxis, which refers to impairment in performing learned skilled motor actions, to imitation as well as to command, referred to as miming. And you see it with communicative gestures and transitive gestures, such as waving and things like that, as well as using tools, such as using a hammer. And so we examine it with gestures to command, imitation, and tool use. So starting back around 20 years ago, we start to look at praxis in detail in autism. We adapted Ken Heilman and Leslie Gonzalez-Rothy's detailed examination that was developed for Florida Praxia Battery. It was developed for adults, for children, and sort of just going to give you one little quick example here. Oh, that's not what I meant to do. That shows this. So here's a typically developing child first erasing a chalkboard, being asked to erase a chalkboard, and here's a child with autism. And you can see, first of all, this is what it's classically referred to as a body part for tool error, or it could be that just the way that he's relating to the eraser is abnormal, but probably a body part for tool error. But there's a very inconsistent relation also to the imagined chalkboard, as well as other abnormalities. So we did these sort of video recordings and an analysis of it, and have reported in a series of papers with fairly non-overlapping samples. Now, I should say that all these studies that I'm going to talk about are generally school-age children, 6 or 8 to 12 years of age, pre-pubertal, and generally IQs of above 70 or so. And I understand that that's sort of a focused population here, and I could have a discussion after about sort of the benefits and downsides of that. But we are looking now to translate into broader populations. So first in 2006, in Journal of International Neuropsychological Society reported this impairments in gestures of command to imitation and with tool use in children with autism, and replicated this a number of times, including in a paper in 2012, where we not only showed that the impairment was present in the upper right corner in praxis in children with autism compared to typically developing children, but also compared to children with ADHD. So we see that that there may be some specificity, this evidence of specificity for dyspraxia and autism. And furthermore, in the bottom right-hand corner, you see something called the PANS that my mentor, Martha Denkla, who, by the way, was a mentee of Norm Geschwind, related to the person, one of the people who invited me here very kindly, and the PANAS, which is a motor battery of basic motor control developed for children. And there you see that higher scores are worse there, that children with ASD and children with ADHD both perform worse than the typically developing children. So whereas we see these basic motor control impairments in both autism and ADHD as shown in the bottom right-hand corner, we only see the dyspraxia in the children with autism. Now, there's a little bit of impairment in the children with ADHD, but not nearly to the degree that we see in children with autism. So then this brings us to sort of talking about the brain basis and neurology of praxis. And this is something adapted from Marco Iacoboni, who's, I think, out there somewhere in the Zoom world. And what I like to point out is, and this is obviously Marco works a lot with mirror neuron system and the notion of imitation. And the core of what is that mirror neuron system is a parietal frontal interaction, which really is what defines praxis. So typically left dominant in the parietal cortex, you sort of store these spatial temporal representations, these perceptual representations. So Hugo Liepmann observed, and others since, that if you lesion the parietal cortex, you not only lose the ability to perform these skill gestures, you lose the ability to recognize other people performing these skill gestures. So if you ask somebody like to show me, if you show these individuals, somebody like doing this kind of motion, like hammering a nail, or this kind of motion, like sawing a table, they won't be able to recognize it, in addition to not being able to perform it. However, if you lesion the frontal or the connections between parietal and frontal, you lose only the ability to perform and retain this ability, this sort of perceptual awareness. Now, that's interesting from an acquired standpoint, but we have to think about development here. And so from a developmental framework, it's unlikely that these things are separable. What's going on is that when you learn these skills, you're forming the perceptual and motor model at the same time, an action model at the same time. And it's sort of an interactive system. So from a developmental framework, there's this idea that you're forming these internal models of action or behavior, and that those models are important and crucial, not only for developing the ability to perform these actions, but also understanding the actions of others. So this led us to sort of say, well, we should assess this in children with autism. Do they have this awareness of these kind of gestures? So we, again, went to Leslie Gonzalez-Rothy and Ken Heilman, actually directly to Leslie, and got the postural knowledge assessment that they do in adults with apraxia to assess this. So you have something like this, you have a multiple choice, you have to choose, obviously, and the answer here is A for hammering a nail. And so we did this in children with autism and children with ADHD. And sure enough, I found, again, that the children with ADHD basically performed comparable with the typically developing children, whereas the children with autism performed worse than both groups. So it suggested that the children with autism, that they show this impaired ability in both performing skill gestures as well as identifying others' gestures. Now, a number of you are, pretty much everybody, I think, in this audience works with children with autism. So you see where I'm going with this, I'm sure, is that you have this interrelationship between motor skill and motor awareness. And that is very parallel to what we see in children with autism when you think about social skills. They have problems with executing social skills, but they in turn also often have problems with what's called social cognition or social awareness, understanding those skills as performed by others. And we've seen evidence to support this. So you see associations between the measured skill performance of dyspraxia with measures from the ADOS, and we've seen this with the SRS as well, and other kinds of measures and other people have observed these kinds of associations between dyspraxia and the core features of autism. And I would submit that this is sort of what sort of contributes to what you kind of refer to as the shake hand diagnosis of autism. What I mean by this is, and let's think about, for example, this population of higher functioning children with autism. There is, the gold standard, and Kathy Lord is part of this working group, is the ADOS, but really the gold standard is clinician impression. And the clinician impression, where does that come from? Of course, it comes from a careful history and detailed, discussion with parents and observation of the child. But there is this sort of sense that people get a lot, oftentimes, in interacting with individuals of sort of like, what is it that that I feel like this person feels like they have autism, or this child or adult. And I would submit that this is sort of a big part of it, is that you're sort of observing their actions and how they interact in that moment, even if it's sort of subtle and not really codified, although then Kathy did a brilliant job of doing so in developing the ADOS. So this sort of basis of examining and defining dyspraxia in autism then led to a couple of things. The first notion was, well, again, this is a developmental process. And I want to point out, too, it's a particularly distinctly, clearly developmental process. And I want to contrast with ADHD. And for that matter, in language disorder. In both of those cases, you can have acquired lesions that result in behaviors that basically emulate what you see in those developmental processes. Take ADHD, frontal lobe injuries, classically, Phineas Gage isn't Phineas Gage anymore. They can result in ADHD behavior. Now, you wouldn't diagnose them with ADHD because it needs to be an acquired condition, but you diagnose them with, a post-traumatic brain injury. But the behavior is very similar. Whereas in autism, you really don't see acquired lesion-based autism very much, if at all, where it's really clearly this uniquely developmental process by which we really should be thinking about how people develop these skills. And I do want to point out that they are called social skills and communicative skills. They're not social facts that you memorize, although this is something that people with autism sometimes do. And so in thinking about this, I want to just sort of quickly discuss this sort of general, very, simplified schematic framework of the fact that there is this contrast between declarative and procedural learning. So procedural learning is kind of what I'm going to be talking about, referring to the ability to acquire skills. It tends to be, quote unquote, like riding a bike. You learn them, they can stay fixed. And that could be good because you can pick up a bike after 20 years of not riding it and still ride a bike. But on the other hand, that could be problematic because they can be difficult to change and modify. This is in contrast to declarative, and procedural learning tends to be these kind of motor, premotor systems that I've discussed. And I'll discuss some subcortical contributions down the line. Declarative tends to be more hippocampal, medial, temporal based, and then with connections to cerebral cortical regions. And this is sort of conscious, explicit learning of facts. Now, the bad thing about this, and we've all experienced this, we cram for a test, we memorize all this stuff. A week later, we don't remember it. But the good thing is it's flexible. If I tell you that I'm going to give you a test tomorrow, and I tell you that the Declaration of Independence was in fact signed in 1775, and I am giving the test, and I give you the test, and you have to answer it, you can easily say, oh yeah, he told me 1775. I guess that's what I'm going to answer, even though for decades you've had 1776 in your mind. So what about quick discussion of procedural skill learning from sort of a more clinical, mile high level in autism. So the development of social skills does involve learning a series of complex action movement patterns, and sort of discussed that quite a bit already. And in addition to the fact that in learning these patterns, we form internal perceptual motor models, which support both development of performing those actions as well as understanding other social actions. And then interestingly, autistic people often report difficulty with automatically performing social actions and with compensatory use of declarative scripting in order to sort of move through social situations. I do remember once, about 20 years ago, so upon meeting Temple Grandin, she told me that, yes, when I get into a social situation, I actually have to prompt myself to stick out my hand and say hello to somebody. I don't feel like I know how to automatically say hello to somebody. So I use scripts in my mind. And we know that this is quite true. And this is in a lot of ways the basis of one of the most commonly used approaches clinically, which is social skill groups, where you sort of all these things that we sort of implicitly, unconsciously learn. You sit children down, and you teach them in a more sort of clever way. Now, hopefully the goal is that they practice this and it becomes embedded in more procedural based learning. So taking now the step into sort of more detailed analysis of motor learning, the initial approach that we took was looking at motor adaptation, reaching adaptation, which tends to be very cerebellar based, and I'll get into a bigger discussion of that later. But this is Courtney Haswell, and we published this initial paper in Nature Neuroscience, and she was the first author in 1999. And this was in collaboration with Reza Shadmir and his group. I'm not going to go into big detail about this. I don't have time to get into the real weeds of all of exactly how this experiment was set up. But basically, they were moving a robotic arm, but they saw a screen. As you can see, they don't see the arm. They only see the screen where there's a cursor, which can give them the correct information, but also can give them manipulated information. So the overarching thing is that we published a series of 3 papers using this approach. The first one I mentioned in 2009, as well as a follow-up paper, Azawa in 2012, which I'll talk about in the next slide a little bit more, where basically we showed that children with autism versus typically developing children learned reaching actions. We had them learning in this novel force field shown on the right schematically. And then the arm configurations were then rotated, and learning or generalization was assessed in visual and proprioceptive coordinates. It would take me about 5 or 10 minutes to explain all this, so I'm just going to sort of have you trust me that we did that correctly. And what we found was that ASD children compared with TD children showed greater learning from proprioceptive error. So when we looked at how they learned, what happened is that the arm is sort of pushed to side and they have to learn to adapt their movement to hit a target. And when we looked at how they learned performed that adaptive learning, we saw that the children with autism learn to a much greater degree than typically developing children in proprioceptive coordinates. Proprioception refers to the awareness of your body in space. I often like to bring people, I'm not going to in this sort of mixed Zoom live environment and sort of demonstrate a testing for proprioception, but basically it's sort of what allows you to close your eyes and touch your fingers to your nose, like to know where you have spindles in your muscles that constantly feed to your somatosensory cortex and connect to motor cortex intimately and give you a sense of where your body is in space. In contrast, the children with autism learned less from visual coordinates, what they were seeing out in the environment. Further, as I'll show in the next slide, the degree of relative proprioceptive versus visual generalization was associated with measures of social impairment in autism, autistic social impairment. A follow-up paper. So that's this.
Stuart, can I ask a question about that? Just quick.
Sure, Dan.
Yeah, sorry. It's super. So usually when we're testing that, like in trying to separate, cerebellar from others, I mean, proprioception, like the finger to nose, for example, involves you use your eyes, you use vision to help with that as well. But if you close your eyes, right, people lose that. Is that the kind of thing that you do to kind of separate that, so it's clearly separated, the pure proprioceptive versus pure visual?
It's a little, it would take some time to explain exactly how we do this. I will tell you in the second experiment, which sort of more clearly demonstrates it in the Marco experiment, which sort of the schematic is shown here. In both cases, you can see basically we're looking at proprioception versus visual, and you can see the dichotomy that's shown. In that experiment, the classic thing we do is we give them the wrong information. They see the cursor, they feel their arm being pushed, but then the cursor goes straight ahead. What's interesting is that the typically developing children are more affected by that than the children with autism. The children with autism end up sort of discounting what they're seeing visually, that they actually learn better under that circumstance than the.
I mean, could it just be that the sensory information is just kind of overwhelming in a way, is more salient, stronger, et cetera, rather than a true kind of fundamental learning issue?
I can't necessarily dismiss this. You can get into a chicken and egg discussion of this. Is the sensory information overwhelming because they tend to rely overly on proprioceptive feedback from their own internal world and tend to, like, they have a system that's set up that way, and then maybe that then results downstream in the, the external sensory world feeling overwhelming. So I could say, like, so I think you can go round and round with that.
Well, I guess you could relate. You could measure that, right? You could measure both of them and see if there's a strong correlation between the performance and learning and their actual sensory hypersensitivity.
Yes, and we actually have measures that, so it is a great idea. I do think that, and I think it's something we can look at with some more recent experiments we're doing now. Again, though, I don't know that until you get down into infancy, which I will admit is like really crucial. You don't know how to disentangle the cause and effect, so to speak. Association is not causation, basically.
Yeah.
But I think that's an excellent point and great question, so thank you. I want to point out too that, again, we saw these in the original Haswell paper. We reported association with some measures of social, particularly social interaction score from the ADOS, and also crucially in that experiment. In the follow-up paper by Azawa in 2012, we see that target 2 is the visual target. The children with autism learn much less from the visual visual coordinates compared to both typically developing children and children with ADHD, shown in green, and learn much more proprioceptive target compared to both groups as well. So again, it seems like the bias for this proprioceptive versus visual feedback not only correlates with some aspects of autism severity, but appears somewhat specific to autism. I am going to run through now a series of like 5 or 6 slides that basically sort of makes the case it's not just our lab, but there have been a number of other labs around the world that have shown this sort of elevated proprioceptive versus visual bias in autism. So Nancy Minshew in 2004 reported a clinically significant reduction in postural stability only when proprioceptive feedback was inaccurate. Morris in 2015 used posterior neck muscle vibration to produce an illusion of backward trunk movement. They did this in adults, and typically developing adults used vision to counter the vibration-induced illusion, whereas ASD adults did not. Zhang, 2019, this is Cheryl Glazebrook Passmore's group, did this experiment, a reaching experiment, and showed that ASD adults showed longer premotor reaction time and movement times for 3D naturalistic reaches, but when you increase proprioceptive feedback with 2D or a 1D groove path, they perform comparable to typically developing. Gray in 2021 manipulated visual perspective, third-person perspective, and showed it had a large effect on endpoint biases for the typically developing but not autism children. So I'm sorry, and then Lee and Bo had 13 children with ASD and TD perform a visual motor adaptation task, and showed slower adaptation rate in autistic versus typically developing. So I'm not going to get into the detail of all those, but basically, I just want to give the impression that it's not just our lab, but there's been a series of experiments around the world that have really suggested that this bias. So we've also done brain behavior examination of this. So I mentioned that everybody gets this feedback from the proprioceptive feedback. Well, except if you have a spinal cord injury, and there was this very sort of well-known person at Kennedy who came through Kennedy Krieger who had such a lesion and actually taught himself to walk and then run. And he did so by looking at his feet and learning through vision because he had no proprioceptive feedback. Very interesting and fascinating story. And they did functional brain imaging, resting state connectivity imaging on him, and showed that he had much stronger visual motor connectivity. So we said, well, let's look at children with autism taking the same approach. And Mary Beth Nebel did this excellent study where we defined using independent component analysis and intrinsic resting state fMRI, motor and visual components. So 2 motor and 3 visual. I'm not going to do details about how we determined that, but basically we saw then 6 combinations. There was a particular combination that was reduced in autism. And the really crucial thing I want to point out in this paper is we replicated this. This is in our sample, and then we replicated it in a sample from Abide. It was actually Kathy Lord at the time in Michigan State sample, and we replicated it and showed the exact same pattern. In fact, this one is from what I'm showing here is the slide from the Michigan State Abide cohort. Furthermore, we found a strong correlation between this visual motor connectivity pattern and SRS score, so that those with lower visual motor connectivity had higher SRS and more autistic phenotype. Okay, so we've talked about motor skill procedural learning and visual motor, this sort of visual motor disintegration that we're seeing with over-reliance on proprioceptive feedback from the internal world, which kind of makes sense heuristically in terms of autism, sort of creating a whole action, perceptual, motor model system that sort of relies more on what's going on internally and less what's going on in the external world. And thinking about the neurology of this, though, there is importance of subcortical input towards this learning, striatal with more reward-based learning input to these systems, and cerebellar with adaptive learning. And I just want to sort of quickly point out that I've had a long-standing interest in the cerebellum. In 1993, I gave a talk on the role of the cerebellum in cognitive function and behavior. So dating back many years. And so we have been looking at cerebellar aspects, and we actually find that children with autism show this anomalous right posterior cerebellar connectivity to the left inferior parietal lobule, which is this key area that's sort of the praxis area, so to speak. And this is something that was done by a postdoc fellow in our lab, Daniel Lidstone, with Mary Beth Nebel, who was a postdoc and now is junior faculty with us. And not only that it correlates with SRS scores, this reduced connectivity. So we think that I'm sort of been staying on the cortical level, but I don't want to dismiss the fact that I do think that subcortical, particularly cerebellar adaptive type learning, might be particularly crucial. There's also this sort of clinical evidence. This is actually, I took this from, like we all go on Google and search out like images like this. This is actually the one that Safari used in like their, what's that called again? the sort of reporting out that they do, the Spectrum, that's it. In the Spectrum article that was about this first article where we showed that when we took the MABC, this sort of motor battery for children, motor assessment battery for children, and we looked at what particularly sort of defined autism there versus both ADHD and typically developing kids. And we found ball catching was more, as much as anything. And we actually, for those who are familiar with the MABC, separated out aiming and catching, because aiming and catching are like peanut butter and jelly to me. They're very different things, but they go together because they're sort of a social game together. And it was really particularly ball catching that distinguished the children with autism from both typically developing and ADHD children. And this is something then that knowing this, Mary Beth Nebo and Becky Landa worked on looking at ball rolling from the AOC, the battery for assessing infants with autism, and showed reduced anticipatory reaching but not gaze response to the ball rolling in infants at high risk for autism. Also, I'll just show this experiment here where we also then, building on this, recently published this study. Dan Lidstone, who I mentioned, postdoctoral fellow, showed we looked at dynamic tracking of a visual target and showed it was impaired in children with ASD, not only compared to typically developing children, but also ADHD, as well as fetal alcohol syndrome children. So here, what you had to do is sort of press this grip force with your thumb and index finger and either hold it statically, which you're seeing on the left-hand side, on the far right in the graph, you see the static condition, and just hold it there. And this is something that also Matt Mosconi and others in Kansas have worked on. Or alternatively, it moves up and down like this sort of rocket ship-like game, and you have to track it. And it was when it became dynamic, sort of like ball catching, like social interaction, that's when the children with autism really showed impairment. So we have things like ball catching, but then we also, dynamic visual motor integration is certainly crucial to imitation and to, naturalistic visual motor imitation. And so I'm going to focus the rest of the talk on imitation and autism. And there's two basic ways people look at it. Spontaneous imitation. This is mostly looked at in infancy and young children, where you do something and just see if the child naturalistically emulates it. But then we're going to focus on more elicited examination of elicited or visual motor imitation. So, as many of you, I'm sure, are aware, imitation is well known to be crucial for development of social and language skills, in particular those associated with autism spectrum disorder. Have to, of course, shout out Sally Rogers, who's done, really crucial work in this, dating back decades. Justin Williams as well. This has been shown in numerous papers that ASD children imitate others' actions less frequently and with less precision. Impaired imitation has been also shown to be associated with core autism features. And also impaired imitation in autism has been associated with several autism-related constructs, sort of theoretical constructs like empathy, joint attention, understanding others' actions and intentions, theory of mind, social cognition, which is closely related to that, emotion inference, as well as communication. So now we get to the lamppost. Finally, now I'm getting to the lamppost. So assessing imitation. There's limitations in how we've been traditionally assessing it for decades now, in that we have people do it and we either sit there like with, coding right then and there, or we videotape it and then, have people who are trained. It takes a lot of effort to code those. But it can introduce bias, obviously requires all this time and effort. There's these inconsistent operational definitions and varying methods. So there is, as Edwards pointed out in 2014, this need for developing consistent definitions and methodologies. So how do we improve these methods? I will point out in the coming minutes that we can leverage some really key technological advances to do so. So now I get to drunk under the lamppost. So drunk under the lamppost. I'm sure some of you know it. In fact, I know some of you know it because we talked about it last night at dinner. But for those who don't know it, it's the metaphor to me that is the most important metaphor in science and in life generally, frankly. So the story goes is that it's pitch black night. There's no light anywhere, no moonlight, nothing except for a single lamppost. And there's a drunk on all fours under the lamppost. And a cop comes along and he says, what are you doing? Now, in the old classical one, he says, I'm searching for my keys. People don't like to say that anymore. Obviously, you don't don't want the drunk driving. So I'm searching for my wallet. And the cop says, well, okay, fine. Where'd you drop it? And the drunk says, over there by the bar. And the cop says, well, why are you looking here? And the drunk says, because this is where the light is. And this is really what informs all of what we do. I mean, even down to our own sensory construct as human beings, we are always biased towards whatever lampposts we're using. So for years, so actually, I want to tell a little story quick first, but so for assessing the quality and character of human motion, for years, we used nothing but our eyes in real time, years, like eons. But then along came these video cameras of various types, and now, allowing us to assess things through video recordings, as I've discussed. I love this story of the galloping horse problem and the world's first motion picture. There's some French people who did this earlier too, but a bit early as well. But Leland Stanford, California, former governor, very wealthy man and horse enthusiast, and very opinionated and a confident man, as you can see from that picture, was very upset because horses had been painted for years as in the air with legs extended. And he said, that is entirely wrong. I am a horse enthusiast, and this is just all wrong. All the centuries of people painting horses this way is entirely wrong. So he hired this very interesting person, Edwin Muybridge, who I suggest you go read about. Fascinating individual. I think he was associated with John Muir. He took photographs, and was early photographer, and he set up this crazy system for Leland Stanford, because Leland Stanford just threw all this money into it, where he had a horse gallop, and then next to it had another horse gallop with a cart that then triggered the series of still cameras, and got these series of pictures of a horse. And as you can see, the horse in the air when in the air has all four legs tucked under. So Leland Stanford was usually happy. Eventually they had a bad breakup. We don't need to get into that. But Edwin Muybridge then actually used this, and I love that he called it the zoopraxiscope. So he actually sort of said oh, I can set up these series of pictures, and somebody could look through a viewfinder, and I just turn this around in a circle and people could see motion. And so in some ways, this is one of the first motion pictures. He went on to actually really intently use this to study praxis. He did it in humans. Has anybody honest, I'm in Hollywood, seen Holy Motors, the French existentialist film from about a decade ago? It starts off with this great film, but it starts off with these images. But it's but yeah, so he went on to study praxis. But then now in today's day and age, so we've been doing using these approaches to study praxis and imitation for decades now. But now we have this new lamppost, computer vision, where we can actually track motion and assess it more digitally. So we've been applying this to advanced motor imitation as a ASD biomarker, knowing all that we've learned over the last couple of decades. And Bahar Tungchen, who's now in Nottingham, but was with us as a postdoctoral fellow, spearheaded this. We applied computer vision algorithms optimized to assess human motion data, and used sort of this video game, very brief, 1 minute or less video game that's cost effective and accessible, and also importantly, moving forward, scalable. So we can move beyond the research lab and into home and clinic settings, especially now. We had no idea COVID was coming, of course, when we started this. And try to see if we can use it to advance imitation as a phenotypic biomarker and address autism heterogeneity. So there's a team of people who worked on this. I mentioned a number of them already from our own lab, but also in close collaboration with Renee Vidal at the Center for Imaging Science, who's a world-renowned computer vision expert. So this just shows we basically had this avatar, as you can see, and we had this video. They performed a series of movements. It lasts about a minute. And then we have the skeletonized recording of the person performing it. We use this approach that I'm not going to get into too much detail about, dynamic time warping, to assess, to combine measures of temporal distance between the child and the avatar, and the time the child was delayed and the time the child moving in advance of the avatar as well, to come up with a score. And this sort of quickly displays the approach that we take, the methodologic approach to come up with a CAMI score. We also, for this initial study, used human observation coding so that we could sort of validate it. So we did do the sort of old-fashioned way of sitting down And we had this sort of series of things that we looked for in the movements and score them. And sure enough, we found, and this paper was published a couple of years ago, we found, or about a year and a half ago, we found that there was a close association between the computerized assessment of motor imitation scores and the human observation scores across a series of 3 different trials. And importantly as well, then showed that actually CAMI outperformed the human observation code in classifying autism with 87% accuracy, and also that it correlated more robustly with autism severity with the SRS-2 than did the human observation coding, and showed strong test-retest reliability. So we're now moving forward with some things to scale this up as a biomarker. So first of all, broadening to broader autism populations, younger and broader phenotype. Importantly, this was done with, I didn't say with 3D Connect system camera, but we're now using 2D cameras, including more portable, like scalable phone cameras and computer cameras, so we can move into clinic and home settings. We actually did an initial study reporting on association of the 2D cameras and 3D camera that's embedded, both of them are embedded in the Connect system. So we're able to extract the 2D camera data as well. And we showed in this paper that Dan Lidstone published in 2021 that that the CAMI 3D and the CAMI 2D are fairly comparable in distinguishing autism and typically developing, and also they both correlate with SRS-2 at about the same level. We also, the thing about these potential use of these things as biomarkers is it's interesting to distinguish them from typically developing children, but that's not necessarily going to help clinically. Parents come in and their child has, like, the questions are, is it autism, but is it if they're not bringing their child in because everything's healthy, obviously, so there potentially are other diagnoses. The main ones that I have always found are ADHD, DLD, developmental language disorder, or intellectual disability, and anxiety disorders are sort of the main ones that are sort of the differential, as well as comorbid, obviously, with autism. And I will say that that we, as mentioned, I do a lot of ADHD research, so we get this in children with ADHD as well. And we've already in some preliminary data, and this is going to be well, I don't know if it's going to be presented in Stockholm, but we submitted it for Stockholm. That it's but I imagine it will be, and I plan on being there. That just so you know, Connie's monitoring, and the children with autism perform worse than the typically developing children, but and also than the ADHD children, and that the ADHD children perform about comparably to typically developing children. And furthermore, when we look within autism at association between ADHD symptomatology and autism symptomatology, I already showed you it's associated with autism symptomatology, but it's not really associated with ADHD symptomatology across the board. So then we want to also explore brain-based and visual motor integration biomarkers. Showing again Marco Iacoboni's, like, my adapted schematic from Marco, but also Werner 2012 shows us in a little more detail the inferior parietal lobule, IFG, ventral premotor, and the STS, which is important for self-other mapping. And I've sort of not gotten too much into that because I've been focusing on the Praxis system, but also crucial for imitation. And some have theorized maybe the particularly Kevin Pelfrey and others that may be particularly problematic. I also want to point out, because this always gets asked, that look, we're doing this, and I think that there is something about, I think there's evidence and a number of factors that point to me about this sort of real IPL to, parietal to premotor integration that may be particularly important. Also including the fact that, like, this sort of notion of the potential of long range connectivity being abnormal in autism, and short range connectivity perhaps being increased, and proprioceptive motor is as short range just about as it can get. But I can't dismiss the fact that, like, anywhere along this stream from your eye all the way out to your hand, like, is when we're asking people to imitate, that is, the entire stream. And it may be more sensory integration itself, the integration of the proprioceptive and visual, or even the visual itself. And we are trying to move forward with doing some things that help us discern that. I do want to point out that one of the, really important first studies that looked at this was Morella DiPreto study in 2006. and actually did imitation in the scanner and reported reduced activation during imitation observation of small finger movements. And this is in contrast to what we were doing outside the scanner. Now, why? Of course, we know, because the lamppost we're using is fMRI, where you have a very restricted environment where it's hard to examine imitation in a more naturalistic way because of the confined space. It's very sensitive to motion, also noisy and distracting. So what about a new lamppost? We are now moving to collaborate with this great person, Adam Egebrecht at WashU, to apply something called high density diffusion optical tomography, basically FNIRS for those who are familiar, but throughout the brain, the cortex at least, or most of the cortex, and with high density. You can sit in a chair. It allows much more freedom of movement. There's not much noise. Adam's done a series of experiments to show that HDDOT produces fMRI comparable maps of brain function, at least within the cortex. It is limited to the cortex. And we now with Adam, and we actually have this R21 NIMH R21 to apply this. We are now using this in children as young as we're moving down to about 36 months of age and hope to move younger. He has done HDDOT with children as young as 18 months, I should say. In all fairness, this is Adam's child at 18 months. So, but we are now performing these experiments with children as young as that and using HDDOT. I also want to mention that, all right, so we have imitation, but we understand that this may be hard to probe in very young children and wider range. And we may want to sort of really look at visual motor integration itself. So we are working with various collaborators to develop tasks sort of like on tablets, like ball catching and other kind of visual motor tasks to try to look at visual motor integration itself, the process sort of distinct from imitation even, to see and then try to scale these down. We'd like to try to develop these for school-age children initially and then scale them down to younger children so we can look at things like this. I can mention quickly that Jerry Dawson has this sort of bubble popping task. I think there are some limitations to that. I can get into discussion of that if anybody has questions about that, but I think that developing some tasks like that allow us to look at visual motor integration and how it may be abnormal in autism. All right, I know I'm out of time, but I want to beg you for 5 minutes to talk about future directions, including in particular therapeutic implications. So if we think about the fact that if the evidence seems to support that that there is at least a group of children with autism, and of course I recognize the heterogeneity, and this may not be true for all children with autism, but if we can identify those with autism who have this over-reliance on proprioceptive feedback and reduced reliance on visual feedback, we can look to either A, teach motor skills better, such as handwriting or sign language, by playing to the strength. In other words, increasing proprioceptive feedback. The problem is with social skills is that would take a lot. It would take like an exoskeleton, which seems not really probable at this point. So the other option is to work through and try to strengthen the system. That is, we can maybe try to strengthen it starting at a very early age, because and possibly through practice with imitation. So I'm going to skip through the motor skill therapy. There's some evidence to really support this, and we've done a lot of stuff on handwriting. Then I'm going to skip showing handwriting is abnormal in autism, and like thinking about the possibility of using increased proprioceptive feedback to improve writing. Also, Dan Lidstone has developed this Hapticart, which allows where you steer a wheel and then you get proprioceptive or alter proprioceptive visual feedback. So we're starting to look at that in a game-like task in children with autism. But I want to focus on the social skill therapy, where we feel like the way to go may be to strengthen this system, this visual motor integration system. So can we alter the way the autistic brain learns skills so that children with autism are better able to form action models that are based on visual feedback, increase the reliance of visual feedback as opposed to proprioceptive feedback, as this is critical to social learning. So can we increase frontal parietal mirror neuron system connectivity? One way to do it is external. So like you try to ramp up using like non-invasive brain stimulation, NIBS for short, transcranial magnetic stimulation, or TDCS. I'm not really going to get into that too much. I think there's some challenges there, but it's worth thinking about. Or we can do it through behavioral approaches. So one thing we've been thinking about is, can we use Kami to this motor video game to improve ability to imitate other people's actions in natural settings? So we improve visual motor connectivity and hope then if we do it in a game-like setting, that if we can improve this connectivity, then they go out into the real world and system is strengthened and they could rely increasingly on visual feedback from the external world. So we know that slower speed helps improve visual motor integration in children with ASD. Wyatt and Craig showed this. I won't get into the experiment. It's pretty simple, straightforward experiment, but there is some evidence to suggest if you slow things down, the children will get better. You make it less dynamic. So can we use slower speed and repeated practice to enhance imitation and underlying visual motor connectivity? So we would slow the speed and then gradually increase it. So we would do something where like we just slow it down to like 60% speed and then see how children with autism do, and then possibly increase it. You have them do repeated practice with increasing. We have looked at 60% speed, started to look at it, and we do see that the children with autism, everybody improves, the typically developing children and the children with autism, but we can get the children with autism up to about typically developing levels using this approach. So we think that there might be some benefit there. So that's very preliminary, but I hope gives you a sense of the kinds of things we're thinking about with intervention. Generally speaking, and overall, how we're moving from like looking at behavior, looking at brain, and then sort of informing not only how to maybe better identify individuals with autism and cut through the heterogeneity and identify crucial phenotypes based on these kind of motor assessments, but then also potentially inform intervention. So I'm going to turn back to my acknowledgements and thank you again for inviting me and allowing me to speak and giving me a little extra time here. And I'm happy to take any questions.